motivators for my interest and Elise's interest in metagenomics, so she has a great credit is owed to her. So she's been at Rotherham for several years, many years in fact, and with a background in it was biology initially, wasn't it?
Well, I was a microbial geneticist originally.
Oh, okay. Okay, so without further ado, I'll let
Well, thank you very much, Wally. That was a pleasure to be here. I've learned a lot. So I called it digging into the soil metagenome, but it's really just about how we've tried to analyse it. I'm a microbiologist, I'm not a bioinformaticist, I'm not a statistician, and I've been trying very hard to get to grips with how we deal with very large data sets. But soil is a very complex system, and we work, Rothamsted, as I will explain in a minute, is an agricultural research institute, so we work on soil and plants and sustainability, and plant nutrition and crop production. Soil is very important. It grows our plants. It provides and protects plants from disease, but it's a source of pathogens, the bioindicators of sort of soil health there. Nutrient cycling is a major role of soil. Microorganisms contribute a lot to the structure, both fungi and bacteria, adding filamentous stabilization, hyphae and bacterial filaments and exopolysaccharides that glue together particles. It decomposes xenobiotics, so it's like a big sort of clean-up. It's also seen as a gene pool for potentially novel biochemical pathways, and a lot of antibiotics that we use come from soil-dwelling organisms. And it's a major source of gaseous fluxes, some of which have significant greenhouse effects. The soil food web is extremely complicated, and it has all sorts of organisms from bacteria and fungi, and I suppose phage at some level, right up to the mesofauna that graze on it and the animals that interact with it. And it's also subject to different sorts of perturbations, especially in agriculture. So when you hear ecologists talking, they sometimes have a slightly different view of this, but if you work in arable agriculture, you know that a lot of the soils are cultivated or tilled annually. This is a major perturbation. They have quite large amounts of fertilizer added, big inputs of nitrogen, smaller inputs of elements like phosphorus. And then of course, a lot of the crop is taken away, so it's not recycled into the soil, it's removed for food. So it produces stuff and it's taken off-take. And quite a lot of land is grazed by livestock, and this produces, of course, different sorts of input. So animal-associated organisms get deposited unevenly in sort of blocks of feces, and they have a sort of another sort of deposition. And of course it's highly variable at different scales. So there's the landscape scale, field scale as well, that's just an aerial view of Rothamsted. At the sort of clod scale, big clods, and you can see plant detritus. Also at the aggregate scale, you've got microaggregates and larger aggregates here, but they have different surface areas and they'll have different mineral and organic composition. And even at the scale in which microbes live, and I took this from what's it called, Commons image of microbes in soil, that's hyphomicrobium on us, but you can see all the different sort of thing that's around this bacterium that's adhering to what I presume is a lump of organic detritus. So extremely variable environment that they live in. So perhaps it's not surprising that they're highly diverse, and metagenomics has been a boon to soil microbiology because fewer than 1% are culturable, maybe around 1%, but it's extremely abundant. Bacteria are extremely abundant in soil, so there might be 1 billion bacterial cells per gram, and there might be it depends on which definition you use, but there are at least 10,000 species, maybe more, every gram. And the next gram, or the next aggregate, will have some things which are the same and some that are different. So it's very difficult to actually achieve a full identification of everything there. You'll never reach an asymptote on a collector's curve from soil. So, very large metagenome. We'd say, I would argue, greater than anywhere else. I mean, the human genome's tiny by comparison. The Sargasso Sea is pretty small. The human microbiome might be sort of bigger than the human genome, but it's still less than, if you think about, soil as a whole, less than that. Now, why do we work on this at Rothamsted? So I'll introduce that to you. We're a mainly government-funded research institute, Research Council funded. We get money from elsewhere too. And we're probably the oldest agricultural research station in the world because we were founded by an amateur chemist who had patented phosphate fertilizer. I think he originally wanted to be able to show the efficacy of his product, so he set up sort of field testing and experimental plots, and quite quickly recruited a trained chemist who'd been trained in Germany to look at nitrogen as well. So some of the earliest experiments were begun in 1843, which is a long time ago now. And some of those have been running continually ever since. And one of the things that we do is to investigate the impacts of agriculture on soil quality, which is difficult to define but important. And we want to know how land management, fertilizer use and other inputs and climate change, different crops, are going to affect crop production, sustainability. And one of the things which is quite useful, is fairly typical Northern European arable and pasture management. So, we can act as a model for other sorts of soils of similar types. And this is an aerial view of Rothamsted, so these are some of the experimental plots I'll talk about. And we've got our labs near Harpenden here. And there's an old manor house where John Bennett Laws used to live, sort of in the middle. And perhaps important for some of the work on nitrogen, the M1 runs somewhere up here, you can see that, and the M25, which is the major London ring road, somewhere here, and Luton Airport will be somewhere up there. So although we're in quite a rural area, we are subject to deposition from various internal combustion engines, nevertheless. One of the advantages is that we have in the past had a very large soil science influence, and so there's a quite comprehensive soil map available of the soil types at Rothamsted, which is very useful. It's all fairly neutral pH and it's fairly high clay, but I mean there are differences over the land. This is about where the last ice age finished, so it's quite a variable sort of soil types. And we also have metadata, because the people who worked at Rothamsted like collecting data, and they've been collecting it for years. So they've been collecting meteorological data since 1843. So we've got air temperature, rainfall, air and soil temperatures, and the hours of sunshine. So these records sort of exist, so they're there as quite a useful background to things that we may measure. We also have a large archive of samples that have been collected since that time. Now, the soil's dried and milled, so it has a limited utility for microbiological stuff. You can extract DNA from it, and Ian and I have had a look at that. It's not quite the same as DNA from fresh soil, but as our methodologies improve, we may well be able to go back and look at things from the pre-industrial age, from 1843 is pre-industrial. And in fact, they encompass pre to post-industrial times. So in recent years, actually, they found that Rothamsted suffers from a shortage of sulphur because we no longer have pollution from the industrial Midlands falling on us. So they have to add sulphur to the fertiliser to help the crops grow now. And the field sites that we've been focusing on are Broadbalk, this is the original site set up, and it has strips, And it was set up prior to the invention of sort of statistical methods. So it has strips with different fertilizer treatments. And at the end, this is quite important, a bit was left. It had been in cultivation for a few hundred years beforehand as sort of wheat fields. It was a good wheat field. But they stopped ploughing up here. They cut trees down in one bit and let the other grow. And that's now a mature wood, a sort of narrow wood, but it's a wood. And this is now a field, but it was once like this. And crossways, there are different treatments with different amounts of phosphate, herbicides or no herbicides, and fungicides or no fungicides. And although it's extremely valuable because we now have a sort of stabilised, I suppose, almost steady state soils with different levels of inputs, there are problems of replication which referees always beat us with when we try and publish. And it's one of the reasons that Fisher had to invent randomised block design because of the problems analysing this. But we're stuck with it, and it's so valuable that we think we can use it with pseudo-replication and so on. And park grass is another very old experiment, which is grass, because grass was the biofuel of the 19th century. You used it to feed your hay for horses. And this is also to look at the effect of fertilisers to see if that improves plant productivity. And it's subsequently been used for more ecological experiments to look at plant composition. For example, the diversity of plants is reduced if you have higher nitrogen inputs. And then a more recent experiment, which we are very excited about, is called Highfield, which was set up as originally it was a grassland, and they decided after the Second World War to plough up some of it to turn into wheat, so it was under arable cultivation. So we have permanent arable and permanent grass plots here, and it's all replicated. And at the end and up the side here, they left an area that they decided to leave unplanted and keep clear and plough regularly. So it's a bare fallow. It's very unusual to have a bare fallow, but it lets us look at the influence of actually having plants or not. And this, of course, is very useful because there's not much soil without plants on. They try and grow wherever they can. So this is the sort of most recent of the ones that we work on. And we've been looking at mainly with collaborations, because at the beginning it was too expensive. We couldn't afford to get any metagenomics done. It'd be a bit different now. But on Broadbalk, we started a collaboration with Eric Triplett of the University of Florida, who got some NSF money on the Genomic Observatories program, I think that's what it's called, to do amplicon sequencing on a range of plots with different fertilizer and manure inputs, and the grass and wood at the end. And we got about 70,000 16S ribosomal RNA amplicons from this altogether. And the main, the first analysis of that was published last year, beginning of last year. The park grass experiment was the site of the original Terragenome initiative to look at metagenomics in soil, and that was with Tim Vogel from the University of Lyon in France and his colleagues. And they had French government funding to try and get a soil metagenome, so that was only 20 million reads. It doesn't sound very much now, but using 454. But that's also led to publication in 2012, in fact, on here. I'm not going to say much more about park grass. And the Highfield plot, which is the, if you like, possibly the most valuable because it's fully replicated and because it has these extreme differences between plants, no plants and arable production, or grass and permanent cultivation. We now have quite large metatranscriptome, metagenome, and an amplicon data set. And this has been done partly with Janet Jansen, Volker Meyer, and Illumina in a collaboration, and partly with TGAC in Norwich, and David Swarbrick's here today. So the nitrogen cycle is probably the thing that concerns us most in our work. We're interested in other nutrients too, mainly because it's essential for agriculture to use nitrogen, and it's a major, if you like, user of nitrogen, but also a major provider of pollution, so, you know, leaching and whatever. And why do some of these people talk about nitrogen as though it was a nasty pollutant? I can't understand why people. It's a very good reason why it's used in modern agriculture, and that's because of this. These are results from Broadbach. The more nitrogen fertilizer you add, up to a certain point, you get a linear increase in yield. I mean, that's sort of fairly straightforward. So it's very important. So this is a 10-year mean for the last 10 years on Broadbach of wheat yields. So up to, I don't know, this is almost perfect correlation. Maybe it drops off when you get to the very highest rate. So the normal farm rate is somewhere around here. That's sort of presumably the most economically valid. I imagine that depending on the cost of fertilizer, the cost of wheat, and the cost of fuel, the sort of point at which these are more economic will change a bit. But it's quite clear that the more fertilizer nitrogen you add, the better your yields. Now, the nitrogen cycle is also fairly well established, although there's a lot we don't know. So nitrogen is lots of it in the atmosphere. It's 80% nitrogen gas, but that's very stable. Not much of it gets into the sort of ecosystem. And before chemical fertilizers came along, the only way it got in was through biological nitrogen fixation, which is done only by bacteria and archaea. So the NIFH is an enzyme responsible for fixation. I'm not going to talk about this very much. I'll go through it a bit, but this is obviously important, probably more important in natural ecosystems, because at the moment we use a lot of artificial fertilizer, but that will probably change in the future when it becomes too expensive to make it and fossil fuels run out. The ammonia, that's the product, taken up into organic nitrogen and put back into the sort of soil nitrogen pool, and nitrifying bacteria oxidize that ammonia to first nitrite and then nitrate. So that's nitrification, and there are a few other routes by which it cycles, but this is an important cycle. It's important because ammonia isn't very mobile in soil. It's charged, and it doesn't. Plants can take it up, but it doesn't move very far in soil. Nitrate is very mobile. It gets washed out, so it gets lost from agricultural systems, but it also gets to roots, so it gets to gets to plants more easily. So this is quite important to get the balance of this cycle right. If you just put nitrate in, it can wash out very quickly, and it won't be taken up by the plants all at once. If you put just ammonia in, it may not get to the plants when they need it. So a lot of places other than Northern Europe use urea as a nitrogen fertilizer. I think it depends on what the factor is. There's just historical reasons. It's where factories have developed. But urea quickly becomes ammonia. So this is still important. So the next step is the reduction of nitrate, dissimilatory reduction of nitrate to nitrite, and then nitric and nitrous oxide is denitrification. And we know the genes involved in this step. I'll talk about them briefly in a bit. And nitrous oxide is a really important greenhouse gas, and agricultural soils are a major source of this. But some organisms that have the denitrification pathway also have NOS-Z, so nitrous oxide reductase, which results in nitrogen, which, although it's a waste of nitrogen fertilizer, it doesn't damage the environment if it's evolved. So it's sort of key whether this gene is present is quite important. So that's the groups of organisms involved: fixers, nitrifiers and denitrifiers. And just briefly, fixation, we know quite a lot about that. I'm not going to talk about it much, but it's particularly important in agriculture with legumes. I mean, peas aren't very important, that's just an example, but in grassland systems, clover is an important nitrogen fixing legume, so rhizobium and the root nodules fix nitrogen, and in agricultural systems elsewhere, grains like soybean are extremely important, and often they're inoculated with the rhizobium to form nodules because they're not present in the soil. So the sorts of questions we want to answer which bacteria and archaea are active at any one time, and how significant is it in fertilised fields? Probably not. But perhaps more importantly, are we reducing the abundance of some of these by using fertilisers in case we want to go back to pasture or to rely on legumes? So we want to know if we're losing anything long term. The nitrifiers, that's the organisms that convert ammonia to nitrate. We know they're ubiquitous, they're always present, the functions are always there. And we know that both bacteria and archaea are able to do this. And we don't know much about them because they're really hard to culture and really slow growing. And we also know that the bacteria and archaea have very different and distinct genes for ammonium monooxygenase, the enzyme involved. But importantly, there are enzyme inhibitors which are used in agriculture to inhibit nitrification and slow down that step, and they can be very important in trying to regulate the conversion of ammonia to nitrate and therefore the nitrate supply to your crop. And we know there have been some reports recently that bacteria and archaea differ significantly in their susceptibility to these inhibitors. So if the archaea are responsible for nitrification, the inhibitors that are used won't do anything. So there's very little known about this. So that's another sort of question we have. Now we know something about what they look like because in the olden days at Rothamsted, when scientists didn't have lots of reports to write and lots of time on their hands, they could spend 6 months culturing these incredibly slow growing organisms on very difficult conditions. And they actually got, these are old electron micrographs done at Rothamsted that I found in someone's old research book of some of the organisms involved. They sort of got names, and just there are luckily genome sequences for some of these because there have been cultures made. And this one is a nitrite, it's a different group that oxidize nitrite to nitrates. They seem to be fairly ubiquitous too. I'm not going to say much about them. That's not our picture. That's from somewhere else. But the difference between these two groups, so I should say that the sort of second step, which I didn't really go into, is probably that ubiquitous organisms. There's no evidence that they have this interesting two-group structure. So bacteria, we know they're less abundant, and we know their enzyme, the ammonium monooxygenase, has a lower affinity for ammonia. So it actually takes it up efficiently only when there's a lot of ammonia in the soil. And they're strict autotrophs. They fix carbon dioxide. They don't use anything else for energy. And the archaea are more abundant, and they have a high affinity for ammonia, so they only use very low levels of it. But some of them are oligotrophic and mixotrophic, and they can use urea as a nitrogen source, and they can use organic carbon. So they're rather different, and we're only just starting to find out about them. So, we want to know how they both contribute to nitrification, and we want to know if we can manage this. It could be quite important if we want to, if one or the other group is more important. And also we want to know about inhibitors. And denitrification, we know dissimilatory reduction of nitrates confined to bacteria and archaea, although other things have nitrate reductases. Dissimilatory enzyme is only in these bacteria and archaea. And it's very common, but it's very curious because there are two alternative enzymes. And the final enzyme, NOS-Z, that converts the damaging nitrous oxide to innocuous nitrogen isn't always present, and we don't know why. These are very different enzymes. I have no idea why one organism has one and another has another. It seems to be a fairly ubiquitous activity, so when the conditions are right in soil, denitrification happens, and that's when it's warm, it's wet, and there's organic matter and nitrogen present. Then you get denitrification, which is nitrous oxide emissions. So I'll just show that. Yeah. So for instance, on Broadbrook, we can measure nitrous oxide emissions over the year. So if there's no nitrogen added, you don't get very much, and there's a small amount you get less, and a lot of nitrogen you get more. If there's farmyard manure added, which improves the soil structure, even though there's quite a lot of nitrogen, there's less nitrous oxide emissions, and for reasons we don't understand, the woodlands have very high emissions. So we're not quite sure why that is. So we want to know why these two, NrK is a cytochrome CD1, a copper containing thing, and NrS is a cytochrome based enzyme. The significance of one or the other isn't clear at all. And we want to know whether land management can optimise the presence of organisms carrying this third last step in the pathway, the one that allows nitrogen rather than nitrous oxide to be lost. So there's sort of questions we want to know. And the initial data set that we looked at, in fact with Eric Triplett, we had lots and lots of qPCR. This is pre-metagenomics on broad bulk, and that showed, you might expect, that there's very high abundance of bacteria and quite a lot of archaea, not as many as bacteria, but getting there. The bacterial ammonium monooxygenase was much less abundant, except for where there were high nitrogen fertilizer levels. And at that point, it became more abundant or similar to the archaea, which seemed to be generally more abundant. The other thing that we noticed was that in the uncultivated plots, the archaea seemed to be much less abundant. And subsequently, we did a lot of amplicon sequencing, and it's just a principal coordinate analysis I did to have a look at the data on this. You can see that the amplicon from the untilled, the grass and wood plots, they separate from all of the sort of ploughed soils, whatever happens to them. There's sort of some separation of the farmyard manure versus the other ones, but the ones without fertiliser seem to have very similar overall general amplicon abundance and presence compared. So if I used the relate function to look at what was responsible for this, and the main organism that I knew about that was responsible for this separation is Bradyrhizobium. If you we plotted the numbers of Bradyrhizobium and ammonium oxidizing archaea, Candidatus nitrosphaera, which is the main player on our soil, and Nitrosospira, which is the main bacterial nitrifier, you see that the Candidatus nitrosphaera is fairly high almost everywhere, but actually drops in the untilled soils, the grass and wood, and the Bradyrhizobium is much higher, so we're really seeing that effect. The Nitrosospira, which is bacterial, seems to have rather different distributions. That seems to increase as you increase nitrogen levels. So that was the basis of a publication last year. And in fact, if you actually try and correlate this, you can see that the principal archaeal nitrifier, which is Candidatus nitrososphaera, so it's not correlated with an N input, whereas Nitrosospira, the principal bacterial nitrifier at genus level, is very strongly correlated. And that's probably because these levels are all, even with no nitrogen added, probably way above what Candidatus nitrososphaera needs to grow. It can use much lower abundance, so it's irrelevant to its growth. Whereas Nitrosospira relies on the presence of this. So we compared quantitative PCR first, because that was easy to do, for bacterial and archaeal ammonium monooxygenase on the high field plot that has arable, bare fallow, that's ploughed but no plants and no fertiliser, and grass, and compared that to broad bulk using a plot with a similar nitrogen input to this, one with no inputs at all. It has got wheat but hasn't got any fertilisers added, and actually it's very similar distribution. So these two groups are distributing very similarly over the different experimental plots. And looking at amplicons, said that we see this actually increase. I don't know how significant this is, because you saw in the correlation there wasn't really any relationship, but it's much higher on both the tilled plots, then this is on grass, whereas the bacterial ammonium oxidizer is higher, where you've got nitrogen fertilizer added. And on high field, found the same thing, so you can't see it, so low on these two, you can't see it. So these were using different analytical methods, or they're probably similar, but it's giving very similar results using two different methods. So we want to look at this. Obviously, we've got now some valuable data, and as I said, we've got large metagenomic and metatranscriptomic data set from high field. We want to look at this in more detail so we can actually get a handle on the genes we're interested in. So with Elisa Loza, my colleagues in Clark and Andy Neal, who are in the audience, have developed a pipeline to look at functional genes and start to pull things out of here. And decided to actually base it on reference proteins because we know what the enzymes that we're interested in are. We know quite a lot about ammonium monooxygenase. There are lots of sequences in the database. And we have them from archaea and from bacteria. So I should say the candidatus function for Nitrososphaera means it hasn't been grown in pure culture, so it can't officially be made a species or designated as a species, but it can be grown in fairly pure culture, and there's a gene sequence available, so that's why we have information from it. Anyway, so the reference proteins are found and aligned and then retrieved to make a reference tree, and then back translated and aligned. So the DNA sequences are back translated in the right line, and then they're blasted against the metagenome or transcriptome, and then they're placed on a phylogenetic reference tree, and this creates blobs showing the relative abundance. So this uses a hidden Markov model, and that uses p-place. And if you want more details of this, you should discuss it with my colleagues rather than me. So the output looks like this, and it's really quite exciting. So the archaea, so that they're more abundant, we see different blobs in different. This is an archaeal reference tree. So these are archaea that are known to archaeal groups, and that's bacterial. So we have some. This is the arable soil. This is the fallow soil, and that's the grassland. So there are many more on the arable soil, 1,107 sequences in this biggest blob. They come from different organisms. There are about half that number on the bare fallow soil, and they also come from, maybe they're the same, I'm not completely sure from this. And the grassland seems to have slightly different representation. And using this method, it's quite hard to see where the bacterial ammonium monooxygenase. I would have to look at these in a higher resolution, but there are, if you look carefully, you can see spots around here, but there are only tens of numbers here rather than thousands to hundreds, so there are rather fewer, 28 too, written here, it's the total number, so it's actually quite hard to see them. But if you plot them in as principal coordinates, you can see using that data retrieved from the blobs that there is a tight grouping of the grassland. Some of the bare fallow ones seem to share sequence identity and abundance with the grassland. And the arable tends to look a bit different. For the bacteria, where there are not very many numbers, but the archaea, you can see a sort of grouping according to where they come from. And if we look at the communities in a different way, so we compare what we got from the amplicons, which I already showed, which showed more in the arable, and bare fallow is probably not significantly different for the candidatus nitrososphaera. And the metatranscriptome. Now, the metatranscriptome was initially analysed using Megan 5, and David Swarbrick at TGAC sent it to us with the initial analysis. So you can see that we have the metatranscriptome is most abundant, or Nitrososphaera are most active, I should say, in the arable soil. There's more nitrogen. And they're also in the bare fallow and not very active in the grass. And the bacteria seem to follow. Using our new pipeline here, there's actually quite a similar result, although there seems to be more archaeal, relatively more archaeal activity than bacterial. So it'd be interesting to compare exactly what this means, but we think we've got a more accurate way of identifying these. And the metagenome, which we've only done a preliminary analysis of, the distribution looks a little bit different. So we see perhaps what we expect, the highest number of bacterial ammonium oxidizer genes in the arable soil, and fewer in the bare fallow, and then fewer in the grass, and that probably follows nitrogen availability. But for the archaea, I'm not sure if this is statistically significantly different, but again, fewer in the grass. So there are probably reasons for this. There's more overall abundance of things in the grasslands. So, I'm not quite sure how this compares yet, but this is the beginning of being able to actually make sense of these functionally very important genes in an agricultural setting. So this is the beginning. We're hoping that we're going to be able to do better analyses, more analyses, and do some functional tests in microcosms where we actually see if what we think is active when we add nitrogen, see what the effects of inhibitors and such like are. But this is the first step, and this has all been retrieved from our soil metagenomes. So, with that, thank you for listening. And I should say I acknowledge my colleagues at the start, so I wasn't going to put that at the end as well.
Okay, questions?
Well, this is a slightly mean question, which is to suggest there might have been, at least for one part of your talk, a more efficient way of doing things, because so you did full metagenomes and then mapped onto the MOA genes, right? You could alternatively just have amplified up the MOA and sequenced that directly, and it would have been much more sort of cost effective. Which is a mean question, so I'm sorry.
It might have been, but one of the reasons for doing this is that we're afraid that we may be missing. I mean, if you do amplifications, you're using a priori knowledge of sequences that you assume are present. And there may be things that are present that look similar, but actually wouldn't amplify with the PCR primers you use for quantitative PCR. And one of the reasons that we used our approach was to find out if we're actually using appropriate primers, because that's probably a second step in a lot of this, to go and do a more accurate sort of assessment of the relative abundance of things. So, if you like, we used quantitative PCR with the best primers that we had at the beginning. We have done for the denitrifiers, which I didn't really talk about. We did a lot of sequencing for them. But we think we miss things. We think we miss things because they're both quite broad groups, and the primers that are sort of in general use may not be completely appropriate.
Yes, that's a fair point. And even if you have got primers that will pick up all the groups, then you'll get biases associated with different groups. Now, we did a study of something like 50 or so soils, amplifying up the archaeal MOA gene, which we then 454 sequenced. That was with Graham Nichols.
Yes, I know him.
Yeah. So we saw similar patterns to you, though, in that we saw pH was the most important for
I mean, well, that was mostly in grassland, wasn't it?
No, we had grassland, forest, arable, moorland, I think.
I mean, that's a sort of broader approach across a range of things, and we focus on arable soils with some grassland thrown in for good measure. So we're looking at slightly different things, and I'm obviously aware of the work that's being done, and some of the sequence information comes from that.
I guess it would be interesting to see whether you did find any groups that we didn't find using the metagenomics.
Absolutely.
Right?
I mean, this is one of the reasons, the rationales behind our approach to try and find things that may not, that we should be able to capture wider diversity that way.
I can ask a question. Your focus is very much on genes rather than individual microbial species. Do you have a feeling that if you were to go for at least sort of partial assemblies, would that give you any more?
I don't really know. I mean, you see the soil community as a system where the genes are probably more important than the genomes of the players involved. There are some things that would be nice to join up. For instance, the presence of nitrous oxide reductase gene with the nitrite reductase. So it'd be nice to know if those occur and which organisms have both. So there are things that we'd like to match up, but it doesn't seem to be primary importance to get. We're not, for this work, we're not trying to extract organisms, isolate organisms for particular functions. Some of this sort of work is to try and find out who you want to get out to do things for you. We just want to know what's working in there. So the answer is probably no.
But at least, yeah.
Oh, no.
Sorry, but I mean, at least if you do collect the metagenomic data, then if people change their mind about what's interesting, then you have the data. But
Yes.
if you've just done amplicon sequencing,
No, that's true. I mean, we've been doing amplicon sequencing, as have a lot of soil microbiologists, for a long time, and we've always felt constrained that it doesn't give us a full picture. In fact, it does seem to match fairly well with what we're finding, but we didn't know that when we began.
Penny, the difference between the arable treatment and the other ones is clear in terms of groups taking place in the nitrogen cycling. But for you, how long does it take to see these changes? I mean, over time, do you think that it's a matter of few years, or it's a building shaping the community over decades, or
We think
centuries?
it probably happens quite quickly when you change land use. So we do have some conversion experiments on the high field plots where we've changed, for instance, long-term bare fallow into arable production or grass, and they're changing quite fast. But I think the answer would be some things will change very slowly or not at all because it doesn't really matter, and other things change quite fast. So there are a lot of different organisms present. So I think both things probably happen alongside each other.
Penny, what do you think is the there's this fantastic resource of these long-term experiments which have now been running for 170 years, and you mentioned that you have samples from soil way back then, and there is in principle the possibility of getting the microbial DNA out of those things. Do you think they're going to be of good enough, or maybe you've already done it, will they be of good enough quality to actually infer a kind of some kind of drift in the pool of microbia in these samples over time?
There might be. I mean, we did try before we had sort of full metagenomics. We actually used quantitative PCR for that, and showed that apparently, for example, the number of fluorescent pseudomonas in the oldest samples was similar to recent ones. So the DNA had survived, even though the organisms couldn't be cultured. The trouble is that you can never be certain. I mean, I think you probably need quite specific questions to answer, and I think that the DNA will degrade at different rates, and I'm sure that the different G+C content has some sort of effect on that, and probably where the organisms were in the soil structure, depending on whether they're stabilised. The DNA that they release is stabilised or not. So we're not sure. We'd like to look at it. It's something that that's why I showed the archive, something we have in mind. Yeah. So Elisa and George.
Elisa?
I noticed that in your last slide there is a slight discrepancy between the distribution for bacteria and archaea that you get under amplicon analysis and metagenomic analysis. I wonder if you think that this is only due to the multiple COP16S copies issue, or is there anything else?
No, the last slide I showed was based on primers for the ammonium monooxygenase, which is a single copy enzyme. So I think it's probably it's primer bias, which is the kind of problem that, you know, we're probably overestimating or underestimating. I think that's the reason why. That's right. I should have pointed this out in response to the other question.
Just out of curiosity, in terms of how are the soil samples actually being stored for up to 170 years?
They're air dried, mostly milled, not all milled, and kept in jars. And in the past, they're sealed, but not they're hermetically sealed. And in the past, people have gone into them without gloves. So some of them. So when we tried to culture things from them, we did find, you know, something like a Staph aureus or something growing. We looked at most of the things were sort of spore formers that could grow, but there was one thing that had probably recently, in the last 10 years, come from someone's dirty hands. So it's more carefully curated now than it was in the past, because soil scientists don't think about microbiology that much.
How big are the samples?
They vary. So some of the jars are a few kilos. So some of them are quite big, and some of them are very small. So some of the older ones are quite small. So, you know, it's a fairly some of the years there was very little. It's not been collected every year, and there have been some gaps in collection, and there are times when things have changed, but there's quite a good Cronus sequence, and there are reasonably large collections. So, you know, it's easy to get permission to take grams out, but not kilos. So we have to get permission to use it. I mean, it's not sort of freely available to us all the time because it's being preserved for posterity, but it does need to be used for things. So.
It would seem that there would be a lot of interest in analysing such data if you were to get it, for looking at sort of population genetics in these things. And there may be a story there to tell. If you concentrate on function, you may miss the fact that certain functions have been taken over by new groups, and would you think that
there might well be. There has been some research done on plant material that was archived, looking at a plant pathogen, and it seemed that over the last few years, I can't remember which wheat pathogen it was, that had the dominant genotype had changed, switched in the last 20 years. But one of our colleagues went back to the very old wheat samples and found that actually this apparently new type had been the dominant type in the 19th century, or the early parts of the 19th century. And I think they decided it was related to the amount of sulfur deposition. So pre-industrial, there was a dominant type of wheat pathogen that changed when there was more sulfur and maybe other things around, and then switched back. So certainly they were able to do it. That was looking, I think, more or less at a SNP, so it was quite a, you know, quite a small marker. But I'm sure that there are things like that can be done. And I think probably the smaller the region that you look at, the safer it will be, if you like.
I mean, now that sequencing costs, you've been held back by sequencing costs. You would have had a lot more information.
Yeah. Now we can get much more sequence, but we can't analyse it. I mean, we have a shortage of bioinformatics support. So I think it's, I can't remember who said this. It may have been Volker right at the beginning, but we were constrained by the cost of sequencing, and now we're probably constrained by the cost of analysis. So we could get a lot more sequence now. It'd be nice to get metagenomic sequence for a lot more of our fields. And we are obviously doing more of those sorts of experiments now, obtaining more data. I just talked about the data sets where we have some analysis.
So there are no further questions. Thank you very much, then.